What you experience as taste is actually the result of multiple sensory systems working together, with the brain acting as the central processor that merges all incoming information into a single unified perception. The tongue plays an important role, but it only provides a small part of the overall picture. Specialized receptor cells on the tongue detect basic chemical categories like sweet, salty, sour, bitter, and umami. These signals are transmitted through cranial nerves to the brainstem and then relayed to higher brain regions. On their own, however, these signals are relatively simple and lack the richness people typically associate with flavor. A major component of what you perceive as taste actually comes from the olfactory system, or your sense of smell. When you chew food, volatile compounds are released and travel up the back of the throat into the nasal cavity, activating olfactory receptors. This process, known as retronasal olfaction, allows the brain to detect a wide array of complex aromas. These smell signals are then integrated with taste signals in regions such as the orbitofrontal cortex, which plays a key role in assigning value and identity to what you're consuming. Without this contribution from smell, foods would seem flat and one-dimensional, which is why everything tastes bland when your nose is congested. Texture and physical sensation also contribute significantly. The somatosensory system detects properties like creaminess, crunchiness, temperature, and even irritation, such as the burn from spicy foods or the cooling effect of menthol. These signals are processed through the trigeminal nerve and add another layer of information that shapes how food is perceived. For example, the same flavor profile can feel completely different depending on whether a food is smooth or crunchy, warm or cold. Vision and expectation further influence taste perception in ways that are often underestimated. The appearance of food, its color, shape, and presentation, can prime the brain to expect certain flavors before the food even enters the mouth. This expectation can directly alter neural activity in taste-related brain areas, effectively changing how the food is experienced. If something looks rich and sweet, the brain prepares for that experience, and the actual perception often shifts to match the expectation. All of these sensory inputs converge in higher-order brain regions that integrate and interpret the data. The brain doesn't just passively receive information, it actively constructs the experience of flavor based on context, past experiences, and current physiological state. Hunger, for instance, can enhance the perceived intensity and pleasantness of food, while satiety can diminish it. This dynamic integration means that taste is not a fixed property of food, but a flexible, brain-driven experience that can vary from moment to moment. When sugar enters the mouth and binds to sweet taste receptors, it initiates more than just a sensory experience. It activates deep neural circuits that are responsible for motivation, reward, and reinforcement. These circuits are centered around dopamine, a neurotransmitter that plays a critical role in driving behavior toward outcomes that the brain interprets as beneficial. The signal begins at the level of the tongue, but quickly transitions into a much more complex process involving the brain's reward system. Once sweet taste receptors are activated, signals are transmitted through neural pathways to areas such as the brainstem and thalamus, eventually reaching the gustatory cortex. At the same time, parallel pathways engage the ventral tegmental area, a key node in the dopamine system. Neurons in this region release dopamine into the nucleus accumbens, which is strongly associated with feelings of reward and the reinforcement of behaviors. This dopamine release does not simply create pleasure, it encodes the importance of the experience and increases the likelihood that the behavior will be repeated in the future. Importantly, dopamine is not just about enjoyment, it is more closely tied to motivation and anticipation. The brain begins to learn that consuming sugar leads to a rewarding outcome, and over time, cues associated with sugar intake can themselves trigger dopamine release. This means that seeing, smelling, or even thinking about sweet foods can activate these circuits before any actual consumption occurs. As a result, cravings can arise not from an immediate energy need, but from learned associations that have been reinforced over repeated exposures. This reinforcement process is highly adaptive in environments where energy sources are scarce, as it encourages the organism to seek out calorie-dense foods. However, in modern settings where sugar is abundant and often combined with fat and salt, the same system can become overactivated. Foods engineered to maximize palatability can produce stronger and more frequent dopamine responses than natural sources of sugar, leading to repeated stimulation of the reward pathway. Over time, this repeated activation can alter the sensitivity of the dopamine system. The brain may begin to reduce receptor availability or responsiveness, meaning that the same amount of sugar produces a weaker reward signal. This creates a cycle where larger quantities or more frequent consumption are needed to achieve the same level of satisfaction. At the same time, everyday foods that do not strongly activate the reward system may become less appealing, shifting preferences toward more intensely stimulating options. The habit-forming nature of sugar consumption is also influenced by how quickly it affects the body. Rapid increases in blood glucose can amplify the association between the behavior and its rewarding outcome, strengthening the neural pathways involved. This is particularly true for highly processed sugars that are absorbed quickly, creating a sharp and noticeable effect that the brain can easily link to the act of consumption. Context plays a significant role as well. Repeatedly consuming sugar in specific environments or emotional states can bind those conditions to the reward experience. Over time, those contexts alone can trigger the urge to seek out sweet foods, even in the absence of hunger. This is how patterns such as stress eating or habitual snacking become deeply ingrained at the neural level. The human brain is inherently biased toward sugar because, at a biological level, sugar represents a fast and efficient source of energy. Glucose is the primary fuel used by the brain, and maintaining a stable supply is critical for survival. Throughout most of human evolution, access to high-calorie foods was inconsistent, and energy scarcity was a constant pressure. As a result, the nervous system evolved mechanisms that strongly favor the detection and consumption of sweet substances whenever they are available. This preference begins very early in life. Even newborns show a clear positive response to sweet tastes, indicating that the attraction to sugar is not learned but built into the nervous system. When sweet compounds activate taste receptors, the brain interprets this as a signal that energy-rich nutrients are present. This triggers not only a pleasant sensory experience, but also a cascade of physiological and behavioral responses that encourage continued consumption. From an evolutionary standpoint, this system was highly adaptive. Natural sources of sugar, such as fruits, were relatively rare and often seasonal. When they were available, consuming them in larger quantities would increase the chances of storing excess energy in the form of glycogen or fat, which could then be used during periods of food scarcity. The brain's reward system was tuned to reinforce this behavior, ensuring that individuals who sought out and consumed sweet foods had a survival advantage. The problem arises in modern environments where sugar is no longer scarce. Instead of being limited to natural sources, sugar is now widely available in highly concentrated and processed forms. These forms deliver a much higher dose of sweetness and energy than anything the brain evolved to handle. As a result, the same biological mechanisms that once promoted survival now drive overconsumption. The brain does not effectively distinguish between natural and refined sources of sugar at the level of reward signaling. Whether the sugar comes from fruit or a processed snack, the activation of sweet taste receptors and subsequent dopamine release can be similar, although the intensity and speed of the response are often greater with refined sugars. This creates a mismatch between the brain's expectations and the modern food environment, leading to a persistent drive to consume more than is necessary. Another important aspect is that the brain prioritizes immediate energy availability over long-term balance. When sugar is consumed, it provides a rapid increase in blood glucose, which the brain interprets as a valuable and urgent resource. This reinforces the behavior even if overall energy stores are already sufficient. In other words, the system is designed to respond to opportunity rather than regulate abundance. Over time, this hardwired preference can shape dietary habits and influence decision-making in subtle ways. The brain continuously evaluates options based on predicted reward value, and sweet foods often rank higher due to their strong biological significance. This can make it more difficult to choose alternatives that are less immediately rewarding, even if they are more beneficial in the long term. This built-in bias toward sugar is not a flaw in the system, but a reflection of the conditions under which the human brain evolved. However, in an environment where high-sugar foods are constantly accessible, it becomes a driving force behind many patterns of overconsumption. When sugar is consumed repeatedly, the body does not remain static in how it responds. Instead, it adapts at both the level of the taste receptors and within the brain's reward circuitry. These changes are part of a broader process known as neural and sensory plasticity, where systems adjust based on the frequency and intensity of stimulation. What begins as a strong and noticeable response to sweetness can gradually shift, requiring more input to achieve the same perception and level of satisfaction. At the level of the tongue, sweet taste is detected by specialized receptors that respond to sugar molecules. With frequent exposure to high levels of sweetness, these receptors can become less sensitive. This doesn't mean they stop working, but the threshold required to activate them increases. As a result, foods that once tasted sufficiently sweet may begin to feel less intense over time. This is why individuals who regularly consume highly sweetened foods often find naturally sweet options like fruits less satisfying. The sensory system has effectively recalibrated to expect a higher level of stimulation. In parallel, similar adaptations occur in the brain's reward pathways. Dopamine signaling plays a central role here, particularly in areas such as the nucleus accumbens. With repeated stimulation from sugar intake, the brain may reduce the availability or responsiveness of dopamine receptors. This process is sometimes referred to as downregulation. The effect is that the same amount of sugar produces a weaker reward signal than it did previously. The experience of pleasure or satisfaction becomes blunted, even though the behavior remains the same. This creates a feedback loop. As the perceived reward decreases, the drive to consume more sugar can increase in an attempt to restore the original level of satisfaction. The brain is essentially trying to compensate for the reduced signal by seeking stronger or more frequent stimulation. Over time, this can lead to patterns where larger quantities of sugar are consumed without delivering the same level of enjoyment that was initially experienced. These adaptations are not limited to conscious perception. They also influence subconscious processes related to motivation and decision-making. The brain continuously evaluates potential rewards and assigns value to different options. When high-sugar foods have been repeatedly paired with strong reward signals, they become prioritized within this system. Even if the subjective pleasure diminishes, the learned association can still drive behavior, making it more likely that those foods will be chosen over alternatives. Another important aspect is the timing and speed of sugar delivery. Highly processed foods often provide sugar in a form that is rapidly absorbed, creating quick spikes in both blood glucose and dopamine signaling. This rapid onset strengthens the association between the act of eating and the resulting reward, making the learning process more robust. Slower, more gradual sources of sugar tend to produce weaker reinforcement signals, which can further bias preferences toward more processed options. Over time, this combination of reduced sensitivity and strengthened behavioral patterns can shift an individual's baseline experience of taste and reward. What once felt like a treat can become a norm, and what was previously satisfying may no longer meet the adjusted expectations of the system. Sugar cravings are not driven by taste and reward alone. They are heavily influenced by hormonal signals that regulate energy balance, hunger, and stress. These hormones act as messengers between the body and the brain, constantly updating neural circuits about the internal state of the organism. Among the most important are insulin, ghrelin, leptin, and cortisol, each of which plays a distinct but interconnected role in shaping the desire for sweet, energy-dense foods. Insulin is released by the pancreas in response to rising blood glucose levels after sugar consumption. Its primary role is to help transport glucose into cells, but it also has significant effects in the brain. Insulin receptors are present in areas involved in reward and appetite regulation, including the hypothalamus and parts of the dopamine system. Under normal conditions, insulin can help suppress appetite and reduce the motivation to continue eating once energy needs are met. However, when insulin signaling becomes less effective, as in insulin resistance, this regulatory effect weakens. The brain may continue to signal a need for energy, even when glucose levels are already elevated, leading to persistent cravings for sugar. Ghrelin, often referred to as the hunger hormone, is released primarily from the stomach and signals the brain to initiate food intake. Its levels tend to rise before meals and fall after eating. Sleep deprivation and irregular eating patterns can elevate ghrelin levels, increasing overall hunger and specifically enhancing the appeal of high-calorie foods. Ghrelin also interacts with the brain's reward system, amplifying dopamine responses and making sweet foods more enticing at both a physiological and psychological level. Leptin acts in opposition to ghrelin. It is released by fat cells and signals the brain that sufficient energy is stored, reducing appetite and promoting satiety. In a well-regulated system, higher leptin levels should decrease the desire for additional food. However, chronic overeating, particularly of high sugar and high fat foods, can lead to leptin resistance. In this state, the brain becomes less responsive to leptin signals, and the perception of fullness is diminished. This can result in continued eating despite adequate or even excessive energy stores, with a tendency to favor foods that strongly activate reward pathways. Cortisol, the primary stress hormone, adds another layer of complexity. It is released in response to physical or psychological stress and has widespread effects on metabolism and behavior. Elevated cortisol levels increase the motivation for quick energy sources, particularly those rich in sugar. At the same time, sugar consumption can temporarily reduce the perception of stress by influencing neural activity in areas related to emotion and threat detection. This creates a reinforcing loop where stress drives sugar intake, and sugar intake provides short-term relief, strengthening the association between the two. These hormonal systems do not operate in isolation. They interact dynamically, with changes in one often influencing the others. For example, sleep deprivation can simultaneously increase ghrelin, decrease leptin, and elevate cortisol, creating a physiological state that strongly biases the individual towards seeking out sugary, calorie-dense foods. Similarly, fluctuations in blood glucose and insulin can alter both hunger signals and reward sensitivity, shaping cravings in real time. The result is that sugar cravings are not simply a matter of preference or self-control. They reflect a coordinated biological response involving multiple hormonal signals that converge on brain systems responsible for motivation and decision-making, continuously adjusting behavior based on internal conditions and external demands. Sugar cravings are not only driven by biology in the moment, they are also shaped by learning and repeated experience over time. The brain is highly efficient at forming associations between behaviors, environments, and outcomes, especially when those outcomes involve strong reward signals. Each time sugar is consumed, particularly in a specific context or emotional state, the brain encodes that experience and links it to surrounding cues. These cues can later trigger cravings even in the absence of hunger or an actual need for energy. This process relies heavily on structures such as the hippocampus and amygdala, which are involved in memory and emotional learning. The hippocampus helps encode the context in which sugar consumption occurs, where you are, what time it is, and what you are doing, while the amygdala attaches emotional significance to the experience. When these regions interact with dopamine-based reward circuits, they strengthen the likelihood that similar situations will prompt the same behavior in the future. Over time, this creates conditioned responses. For example, if someone regularly eats sweets while watching television, the act of sitting down to watch something can become a trigger for craving sugar. Similarly, if sugar is often consumed during periods of stress or boredom, those emotional states alone can activate the urge to seek out sweet foods. These learned associations can become so strong that they operate automatically without conscious awareness. The brain also engages in predictive processing, meaning it anticipates rewards based on past patterns. When a familiar cue is encountered, dopamine can be released in anticipation of sugar consumption, not just after it occurs. This anticipatory response increases motivation and directs attention toward obtaining the expected reward. If the anticipated sugar is not consumed, it can create a sense of dissatisfaction or restlessness, further reinforcing the behavior when it eventually does happen. Importantly, these learned patterns are not fixed. The same neuroplasticity that allows cravings to develop also allows them to be reshaped. When sugar intake is reduced or when new habits are formed, the strength of old associations begins to weaken. This process is sometimes referred to as extinction, where the brain gradually reduces the predictive value of previously learned cues. However, this does not happen instantly. Repeated exposure to the same cues without the expected reward is required for the associations to fade. Introducing new patterns can accelerate this rewiring. Replacing sugar consumption with alternative behaviors, such as drinking water, eating whole foods, or engaging in physical activity, can create new associations that compete with the old ones. Over time, these new pathways can become stronger, reducing the intensity and frequency of cravings triggered by previous habits. Attention and awareness also play a role in this process. By recognizing the specific cues and emotional states that trigger sugar cravings, it becomes possible to interrupt the automatic response and choose a different action. This engages higher-order brain regions, particularly in the prefrontal cortex, which are involved in decision-making and self-regulation. Strengthening these circuits through repeated practice can gradually shift behavior away from cover time. These learned associations can either dominate behavior or be gradually reshaped, depending on how consistently new patterns are introduced. The brain remains adaptable throughout life, and while cravings may feel automatic, they are ultimately the result of repeated reinforcement rather than fixed wiring. By changing the cues, environments, and behaviors linked to sugar intake, it becomes possible to weaken old neural pathways and strengthen new ones that support more stable and intentional choices. In this way, what begins as a conditioned response can be transformed into a more flexible system, where cravings lose their intensity and no longer dictate behavior to the same degree.